Okay, so welcome back, everyone, to the last lecture. So if you remember, yesterday we discussed a simple model for pattern formation in active fluids. If you go back, we had one concentration field, M, myosin, and that was moving around by advection and diffusion. The M field is special because it generates active stresses, and gradients of the active stresses generate flows. The same flows transport the concentration field M. And as we saw and worked out the linear stability in detail yesterday, if you have a system like that and you have a high enough Peclet, which is basically crank up the strength of the activity, then you can go into a pattern state. So the homogeneous state becomes unstable. So depending on whether you have some simple linear reaction or not, if you have a linear reaction, you get a type 1 instability where you get a growth rate which is maximum at a non-zero Q. If you don't have that, then the pattern, the instability occurs at the largest wavelength allowed in the system. Okay, and this we worked out, and we saw that you can get these patternings. And I want to point out and particularly contrast this with Turing reactions, wherein the Turing system had two species, if you remember, U1, U2, like we did in the tutorial as well yesterday. And the essential nonlinearities for pattern forming processes were in the chemical reactions. That's the crucial difference, right? So there was diffusion and there were reactions, and the nonlinearities were really in the chemistry. Unlike that case, for these active patterns, the nonlinearities are only in the advection process and also the saturating. So it's a different kind of a physics there. So rather than having something which is completely driven by local chemistry, here you have something which is coupling to mechanics, and the mechanochemical coupling is what leads to these pattern forming instabilities. So again, to note that if you didn't have this advection, that means you didn't have this driving from the active flows, then this model has nothing other than a homogeneous state. That's the only stable state that there is to it. So if you drive the system out of equilibrium by having these active stresses, then you can have pattern forming instabilities. All right. So one thing I wanted to work out as an example of a model which shows oscillatory patterns, and I want to do that in a little bit of a detail today, and then show you how the same model, just augmented with one more species, can lead to the type 1 or type 2 instabilities, but with the oscillatory component this time. So I'll switch to the board now, and we'll come back to the slides in a few minutes. Okay, so with one regulator of active stress, this is the model that we had. We had one species C. I'll interchangeably use C and M. So we had one species C, which had a advection diffusion evolution. And the advection velocity came from a simple force balance. So this model at the linear stability level can only show stationary pattern forming instabilities. At the nonlinear level, it turns out that this model also has oscillatory instabilities, which is something that we're working on now, and I won't be able to discuss that. So here's the idea. Suppose you had two species. I mean, the analog is something like Turing pattern. And this was something, a bit of a surprising thing when we found this out. Suppose you had two regulators of active stress. And you want to look at the kind of pattern forming instabilities that come up in this system. The motivation, of course, was when we looked at the kind of patterns that come up in the cortex, you saw that the patterns were there, but they were oscillatory. So therefore, it was a natural question to ask, why is it that the patterns are oscillatory? So we wanted to understand oscillatory patterns in active fluids. You can, of course, put in elasticity, you can put in orientation degrees of freedom, and you can have lots of complicated pattern forming equations, which can lead to oscillations. Question was to ask if at some very simple level of chemistry coupled to simple mechanics, you can get oscillations. So here's the idea. Say I have two species, A and I, with the following equations. So I have A, which advex and diffuses, and I, which also advex and diffuses. The only way A and I get coupled, if you look at the equations, their diffusion is diagonal, so there's no cross diffusion. There are no chemical reactions. So the only way that they get coupled is that you say there is a velocity field which is generated by the same active stress model. But this time, the active stress is regulated by both A and I. Does that make sense? So, because these are equations of the continuity form, integral of A over the domain will be a constant A naught, and so will the integral of I over the domain. And these things don't change. So these are input parameters. And so the question is basically trying to ask if I do a stability analysis of the homogeneous state. So there is obviously a homogeneous state like yesterday. So where the concentration fields are A naught, V naught, A naught, I naught. A equal to A naught, I equal to I naught, and V equal to 0. So that's a homogeneous state. I'd like to find out if you did a linear stability analysis, can this system actually undergo some pattern forming instabilities? So just to scale out units and get things in a nice, convenient fashion, I'll just say dA is D and dI is alpha times dA, which is just alpha times D.
Vijay, these A naughts are different from the previous A naughts, right?
A naught depends on what?
No. These A naughts, I naughts are different from the A naught, I naught you wrote in the previous line, right? Because that's the total A naught and I naught.
Yes.
The like the homogeneous set you wrote.
Yes.
Yeah. So these A naughts are some other A naughts, right?
No, it's the same I know. Oh, sorry, I always forget this. You should put the system.
Yeah. Yes. Yeah. Thank you.
Okay, so I have these two diffusion constants. So the ratio of the diffusion constants is I. And as of now, I haven't said anything about the function f. So I want to work this out for a generic f, get some instability conditions, and then come back and interpret those initial conditions. And then maybe see for some particular forms of f, we can say something more. So I go through the usual rigmarole of linear stability analysis. So I'm not going to go through the complete details. So you can surely perturb, so you can define some homogeneous state and define the perturbations about the homogeneous state, and then get the equations, linearized equations for the perturbations. And like we did yesterday, we'll first get the velocity field at wave vector q. So V is 0, so V itself is the perturbation. And this, if you remember from yesterday, will turn out to be essentially the same thing as the following. So you'll get a delta AQ times the derivative, so let's say FA, and delta IQ times Fi. I'll define these things in a minute. divided by eta q square plus gamma. So Fa and Fi are basically the derivatives of the stress regulation function at A0, I0.
Sorry, but like whatever is written after Fa plus isn't visible. Can the capital? Yeah, thanks.
It's not visible in the sense that
the camera hadn't been moved. Now it's
okay.
fine.
So okay, so far so good, the definitions. So just stare at the complete model. Two species, advection diffusion. They get coupled only because they regulate the active stress simultaneously. And the same active stress gradients generates a velocity field, and that's the velocity field that drives both of them. So that's the only coupling between them. There's no cross diffusion, there's no chemical coupling. And I leave it to you to work out. It's very similar to what we've been doing so far. Perturb the system around the homogeneous steady state, linearize the equations, constant coefficients, Fourier transforms, and so get the velocity field in Fourier space. So once you do that, surely you can also do it for the concentration fields. And this time the concentration fields, you can get the, you'll get a coupling matrix now. So you'll be able to write down delta AQ, delta IQ, the time derivative of that as some matrix A times delta AQ, delta. So where the linear stability matrix is what you need to get. So that matrix will turn out to be minus Q square eta by gamma 1, 0, 0 alpha. It's like the dimensionless form. So Q square L square 1, 0, 0 alpha. Yeah, there we go. P Q square L square plus divided by 1 plus Q square L square. Yeah, A naught FA, A naught FI, I naught FA, I naught FI. Where like yesterday, L is this length scale called eta by gamma, and the Peclet number is zeta delta mu. Okay, so everything is well defined inside there. Okay, so if there was no activity, which means the Peclet number was 0, which means zeta delta mu was 0, which means there was no flow, well, then this is a matrix which is just the diffusion part, the decoupled matrix, and that's all nice stable eigenvalues. There's no problem with that at all. Now, if you remember last time when we did the 2 by 2 matrix for the diffusion for the Turing pattern formation as well, the trace and the determinants of A at the Q-dependent function, these are the things that determine the eigenvalues. And once they determine the eigenvalues, then you can work out the conditions where some of them are positive or negative. And that's when you basically get a statement about the pattern forming instabilities. So again, I'll leave the algebra to you. Shouldn't be difficult to do this at all. You can show that, first of all, there will be a, you'll get the following picture. So you can work out the trace and the determinant, and you can work out when the system undergoes a oscillatory instability, and the condition would be that the trace of the matrix at the smallest wave number is positive, and the determinant of the matrix, again at the smallest wave number, is negative. So the answer is that if the Peclet number P is greater than some critical Peclet, or it undergoes at this critical value, which is basically 1 plus alpha times 1 plus 2 pi square, or 2 pi the whole square, 4 pi square L square divided by the system size L square divided by A naught Fi plus I naught Fi. It undergoes at this critical point if Fa is greater than 0 and Fi is less than 0, when alpha is greater than 1. Okay, so that's a summary. I'll just interpret that slowly now. So the math of working out the linear stability, 2 by 2 matrix, eigenvalues, trace, determinant is just a lot of algebra, but can easily be worked out. Please do that. That's homework for you. So go back and think about the physical picture. I have two species, both regulating active stresses. So their regulation of the active stress can be different depending on how A and I control the function F. And the other thing that is different between the two functions is they have different diffusivities. The ratio of the diffusivities is this number, alpha. So you go through the algebra and you find out that the system can undergo a oscillatory instability. That means the system will be such that the real part of the eigenvalues will be positive and the imaginary part will be non-zero. So which means it will become unstable, but it will also oscillate. That happens when the Peclet number is greater than a critical Peclet number and these conditions are satisfied. So what do these conditions mean? Okay, so let's think about the physics of that. So the physics is that when alpha is greater than 1, so which means the two diffusion constants are not the same. The two species diffuse differently. And you also said if the eighth derivative of f is positive and the ith derivative of f is negative. What does that mean? Well, you're saying both A and I regulate the active stress, and Fa and Fi are the derivatives of the function f at the fixed point. So if Fa is positive, it means that A increases the stress, and if Fi is negative, it means I reduces the stress. So an active stress upregulator and an active stress downregulator, with the upregulator diffusing faster than the downregulator. Sounds a lot like a Turing kind of instability, but this is something which is coming completely from mechanical coupling. The system then undergoes an oscillatory instability. Okay, so I'll share the video and show you some pictures. Yeah.
One more condition is that you are saying that we will evaluate trace and determinant at the minimum Q. So why it is so?
So like I said, so this is a system. I'll show you some plots, then you will see. So this is a system which has a conservation law. So which means the growth rates will always be 0 at q equal to 0. So the first instability that will happen will be at the wave number, which is the smallest wave number in the system. So this is 0, and at large q, it must go to 0. Therefore, it will become positive only at the smallest q. So in a discrete system, there'll be a bunch of them, right? And so the q1 is basically 2 pi by L. So remember I forgot the instability. The instability, I think it's type 2, where the wave number, the 0 wave number growth rate is strictly 0 because of a conservation law. And so therefore the system undergoes an instability at the smallest non-zero wave number. Because A and I satisfy continuity equations, the total amount of A and I has to be fixed. So which means the q equal to 0 growth rate cannot be anything other than 0. Okay, so that's the model that we just worked out now. I might have changed notations rather than zeta delta mu caudal C star, it doesn't matter. And so now what you found out was that if I have this condition for the f, that the derivative of f with respect to a is positive, with respect to I is negative, and alpha is positive, then you get a instability. So let's say you choose a function like so, and put some parameter beta, which sort of makes it asymmetric between a and I, and you can actually do a linear stability analysis, and exactly the same pattern that I mentioned comes up. So if you satisfy a Peclet criteria, which is large enough Peclet, but don't satisfy this some other criteria of the diffusion constants being different, then you get a static instability, which means the kind of stationary patterns that we were discussing yesterday. If the Peclet number is large and you satisfy this criteria, you get an oscillatory instability. What does this criteria mean? Just think about it. If you have an active stress upregulator, so beta positive here means it's an upregulator of active stress. Beta greater than 1 means this function increases as A increases. And if beta is greater than 1, this whole thing is negative. So as I increases, C goes down. So if you have an upregulator of active stress and that diffuses faster compared to the downregulator of the active stress, you get spontaneous oscillating patterns. Okay. So in fact, you can look at a numerical simulation. It's very similar to the code that we set up in the tutorial yesterday, but with just one small modification for the active velocity. And you'll get an instability where the patterns are such that the homogeneous state is unstable, and what you get is a pulsating pattern, and that never dies down. So you can simulate this as long as you want, and it never goes down. So there's a whole phase diagram you get, for instance. So there's a Peclet number, and there is this asymmetry parameter. So if your asymmetry parameter is less than 1, then surely you don't satisfy the criteria for these oscillatory patterns, but you can satisfy the criteria for stationary patterns, and you do get stationary patterns. If you satisfy this criteria for oscillatory patterns during like, then you get oscillatory pulsating patterns.
So
yeah,
this beta equal to 0 is kind of special, I guess, right? Because then both A and I, they're like a similar, right? Everything is symmetric.
Yeah, but then it doesn't, I mean, then if you just increase the Peclet number, at some point there'll go some instability, just like what we had in the stationary case with 2 species, and you only get a stationary instability, no oscillations whatsoever. So beta equal to 0, they don't make, I mean, the I and A are not different. I mean, they can be different only in the sense of having different diffusion constants, but that doesn't do any change. There's no qualitative change that happens.
Yeah.
So you undergo a stationary instability. So the criteria is that if beta is less than or equal to 1, mod beta. So therefore beta equal to 0 is one special case. So if mod beta is positive, greater than 1, positive or negative, so then you get this oscillating instability. Okay. Why does it oscillate? I mean, the physics is I mean, you can do a hand waving argument in the following way, that something which upregulates the stress locally will increase the stress. So the black line is the stress, the blue line is the flow velocity. As soon as the stress goes up, remember the velocity field will pull in both of them. So both A and I will come in. But once I comes up, remember I reduces the stress. So locally it will create a destabilization. And so that will push away both the concentration fields. But then A will again try to pull in back. So there's a competition between how fast you move out by diffusion and whether you upregulate or downregulate. And you can write down a nice intuitive picture where you can show that if you satisfy this criteria of fast diffusing upregulator and slow diffusing downregulator, you actually get a nice traveling oscillating pattern. So the physics is that you need to have differential regulation of stress, upregulation, downregulation, and you need to have differential relaxation. Here relaxation is by diffusion. Does that make sense, or is everything flew over the head? No response. So therefore, either everything went in or nothing went in. All right.
What does this mean physically, in the sense that oscillatory pattern?
Can you speak up? I can't. The voice is broken.
Is this pattern being observed in any real world scenario? I mean, what is basically oscillatory pattern?
Yeah, so I mean, one built up this model by actually trying to see if you can get a model for oscillating patterns in a simple scenario with activity. So far, I don't know if there is anything there's an experimental system with this kind of a picture. All the models which show pulsatory patterns in biological problems seem to have some oscillator buried typically in chemistry. I don't know if there is really a molecule which will downregulate the stress. One can cook up some things by assuming there are other, I mean, the cortex is not just myosin, there are lots of other crosslinkers and so on. So some of the crosslinkers can be such that they will actually locally relax the stresses, but no experimental evidence. So you can generalize the model now. I mean, you can ask, hey, should it always have different diffusivities? Like, for example, you had it in the Turing case. Answer is no. So let's say you have both the species with the same diffusivity, D, but put in a simple linear turnover term, but keep the turnover rates different. So you work out the linear stability again, and you get a similar criteria. And this time it says that if you have an upregulator, and that should have a faster turnover rate, Ka, kappa A must be greater than kappa I, if A upregulates the stress and I downregulates the stress. And that thing actually leads to nice traveling waves and loads of patterns as well. So differential relaxation coupled with differential stress regulation is a natural way of getting pulsatory patterns in active fluids. So this is one example where people had asked, if I have non-zero omega C, can you actually start getting oscillating patterns? Okay, I'll flash through some more examples of patterns with this model, but putting in a bit more complication. So I was thinking of looking at some patterns with polarity order parameters and all, but I don't think that will work out. This is probably already going too much.
Sir?
Yeah.
The turnover rate, what do you mean?
So what do I mean by the turnover rate? So if you remember yesterday when we discussed these active patterns, we had the following picture in mind, that there is this actomyosin cortex of cells, which is an active material because of force generation. But the material is not constant. It continuously exchanges material from the surface of the cell to the bulk of the cell. So there is a rate at which material can get bound to the surface or unbound to the surface. That's the turnover rate. That's the rate at which the material gets churned up on the surface. You can also think of it as a rate in which the material, something dies and something gets born. It's like a reaction rate.
So these patterns, whatever we are talking, are there the surface property?
Yes. This was all modeled to look at the actomyosin surface, actomyosin cortex, which is basically a surface thing. So this activity is really happening on the surface of the cell.
So can we say that you said that diffusion and turnover, there are two parameters?
Yes.
So in that case, diffusion or diffusivity was same for the two
Yes.
species.
Yes.
So the turnover rate was different.
That's correct.
So is that either turnover rate or diffusivity?
So you can of course mix up both of them, and you'll get some complicated criteria. The essence is that either you have differential diffusion or differential turnover, and that's enough to give you oscillatory patterns. You can mix up both, and you'll get some more complicated criteria.
Hello.
Yeah.
Hi. No, so I mean, the kind of patterns, what you see, these 2D patterns you showed. So can something be predicted about the patterns from the structure of the eigenvalues, or it's, I mean, oscillatory and all, it's okay, but these patterns are different for these different cases, right? So can anything be predicted about the patterns?
What do you mean? Whether the patterns are stationary, traveling, or hexagonal?
No, yeah. I mean, the kind of honeycomb and these things you showed, right? What kind of patterns emerge? Can that be predicted? Something, the shape and these things? No.
So I didn't understand. So in one dimension, you don't have anything other than this. You can get multiple modes if you put in a turnover, but in 2 dimensions, you can get all these complicated, different spatial patterns, which could also be traveling. Is that what you're asking?
Yes.
Okay. So this has nothing to do with the active stuff. I mean, this is the thing that I was not even planning. I was planning to cover, but I don't think we'll be able to do that at all. So this is basically asking, let's say you go slightly beyond the instability threshold. You can ask what is the nature of the patterns in 1 dimension. Typically, it's some periodic pattern. In 2 dimensions, you can get more complicated things, stripes, rolls, hexagons, and so on. So if you want to decide which pattern gets selected, you have to do something more. So you have to do something called the amplitude equation. And if you write down the amplitude equation for different kinds of patterns, hexagonal patterns, rolls, and so on, you can look at the stability of the amplitude of those patterns. That's why it's called the amplitude equation. And there you can make some statements about which pattern gets selected at which parameter value, close to the stability threshold. That can be done, not been done for these models, but for reaction diffusion models, it's been done a lot. And particularly for Rayleigh-Bénard convection, a lot is known about.
By amplitude, you mean delta A and delta I?
No. So what you can do is these are the amplitudes of the concentration fields. You can assume that the concentration field itself has some, let's say, put in an ansatz like sine QCX, and that'll have some amplitude, right? And so it's basically putting that kind of an ansatz into the equations, working out, writing a consistent theory for this, looking at the normal form, and you can write down an equation for that. You'll get something like Ginzburg-Landau pictures there. So that's valid close to the onset of the instability. So you basically look at one mode truncation, the dominant mode, or few modes. Is that okay?
Okay, thanks.
So the book that I had pointed to in the reading list, Cross and Greenside, that has a lot of discussion on amplitude equations if you are interested. Okay, I'll discuss 1 or 2 more ideas about how this very simple model for active patterns is useful to explain many other cellular phenomena, and I will stop. So I'll come back to this, and I'll switch to the board. I guess I have to unshare. So remember the physics that we are thinking about. So we have a cell, and there is this thin meshwork of polymeric filaments beneath the cell. It's not a static meshwork. It undergoes a constant exchange of materials with the bulk of the cell. So there can be material in the form of monomers here. The monomers polymerize and they get stuck to the surface. And the surface is basically a polymeric gel of randomly cross-linked filaments, actin filaments, and at the same time, loads and loads of molecular motors. Remember, molecular motors are the things that convert energy from ATP into mechanical stresses. So what we've been discussing is a simplified model with one stress regulator, some concentration field C, and this was basically the surface concentration. And you wrote down equations for that. And you also had a surface velocity. And you wrote down something like a Stokes equation for that. And then if you cranked up the Peclet number, basically the strength of the active stresses, we found that there are patterns. So you can have stationary patterns or you could have oscillatory patterns. Now, surely there's the internal constitution of the cell, the cytoplasm. And surely, like we've been discussing about turnover, material when it leaves the surface goes into the cytoplasm, maybe diffuses around and comes back, gets reattached. So we would like to write down simplified physical models for understanding this. So I'll do this in a few minutes for a particular problem of cell polarity, which, if you remember from the very first lecture, is a pattern which gives the cell a kind of a polar nature. So the archetypical example that we had is the case where the cell surface got polarized by one protein on one half, another protein on the other half. And so there is therefore a vectorial polarity that you can associate with the cell. So how about the following picture? So I have a cell, and let's say I have one concentration C, so one molecule C, and this molecule can be in the surface bound state, or it can be in the bulk. And there is a constant exchange between the surface and the bulk. So when it's on the surface, it moves around by diffusion. So what you could do is to write down equations of motion for the surface concentration, which would have some surface diffusion terms. And maybe some reactions when it is on the surface. And similarly, you could have something when it's in the bulk fraction. So there is a surface diffusion, there is a bulk diffusion. And maybe some reactions when it's in the bulk. Assume there are no other sources or sinks for this concentration field. So which means the integral of CS over the surface plus the integral of CB over the bulk of the domain should be a constant. So these are what are called mass conserving systems. Now, you could have a bunch of non-trivial chemical reactions happening here, but you should have the corresponding negative ones happening in the bulk. So such that the integrals of the surface and the bulk concentrations add up to a constant number. Such a model, we'll show you in a few minutes, is very useful for thinking about cell polarity, in particular in the limit where you assume the bulk diffusion is much, much faster compared to the surface diffusion, which is reasonable because in the bulk, 3D diffusion, much faster, surface diffusion is much slower. So you can assume that the bulk concentration is a constant, uniform throughout the bulk. And so you get an effective equation only for the surface concentration fields. And so the dynamics on the surface alone can have non-trivial patterns. For example, a polarity pattern, which the cells use to do some very nice processes happening. So even before going into polarity or looking at multiple species, here's a simpler model that you can think about. So I think about the model where I had one concentration field for the surface, And on the surface, I had a current. So remember, this current is a surface velocity times the surface concentration and some surface diffusion. I say the following, for getting the flow on the surface, I write down a force balance condition. So divergence of the surface stresses are balanced by all the external forces that are coming on the surface. And if you remember, in the simplest approximation, we had said that the external forces, we will model it by a simple frictional drag term. This is the simplest model. A better model is to say that this really comes from the tractional force from the cytoplasm. So you model the cytoplasm as a simple Newtonian viscous liquid. So which means it satisfies the Stokes equation, the Navier-Stokes equation with the inertial terms dropped in the limit of the low Reynolds number. And so therefore you have a equation for the bulk velocity, the cytoplasmic velocity, which is basically some and the bulk velocity field is incompressible. So for people who have done fluid dynamics, this is a standard problem that you would do when you do a first course in fluid dynamics. Navier-Stokes assume viscosity is very, very high. And so therefore you can neglect inertial terms. And if you're looking at the steady state, shear viscosity, you get what's called the Stokes equation. And the three-dimensional velocity field is, to a very good approximation, incompressible. It's an incompressible fluid. So basically it's water, 70% water is what's inside cells. So the surface and the bulk get coupled via the following. So the surface velocity provides a boundary condition for the bulk field on the boundary of the domain. So basically on the surface of the cell. The stresses that come up in the bulk, which is basically some symmetrized version of the gradient of the bulk velocity, they provide a traction force for the surface. So you say the external force is basically the surface value of the total bulk stress tensor dotted with the normal evaluated on the surface. So I'm not saying anything more than the following. I have a sphere, I have a fluid on this inside the sphere, I have an active fluid on the surface of the sphere. The activity of that fluid is regulated by some concentration field Cs. So it will generate flows on the surface. Think of this like Marangoni flows. Surely if there are flows on the surface, they'll drive flows in the bulk. The surface flows provide the boundary condition for the bulk flows. The bulk stresses generate traction forces for the surface flows. This is the equation for the surface flows, very similar to what we wrote yesterday or even today for the two-component active oscillatory systems. And the bulk fluid satisfies a simple Stokes equation. sigma surface, as usual, is basically some eta times grad V symmetrized version plus eta B times divergence of V and the active stress. With the boundary conditions and the traction forces given by this setup. So this system of equations leads to a very interesting set of patterns. I will not go through the linear stability analysis because what it does is to require you to solve the flows inside a sphere. So you'll need what are called vector spherical harmonics. These are the vector analogs of the spherical harmonics that you learn in quantum mechanics when you solve the Laplace equation. There are vectorial versions of that. So you can use them to solve exactly for the flow field given a boundary condition on the surface. And then once you get the flow field, you can evaluate the stress tensor and the drag force, and then put that back into the surface flow equation. That will give you the surface flow, which can be used to move around the surface concentration. So it looks very complicated. At the end of the day, there's only one nonlinearity in this problem, which is the advection nonlinearity. If that were not there, this entire problem is completely linear. There's no pattern forming instabilities at all. So the idea was basically to show you the same idea which I showed in one dimension. A simple advective coupling between flows generated by mechanochemical interactions and concentration fields is enough to generate very non-trivial patterns. So if you look at the equations, that's exactly what I've said. You take the equations for the concentration field on the surface, also put in a turnover term. You can forget that if you don't want. You have the equations for the flow on the surface, which involves the shear viscosity, bulk viscosity, active stresses, and the traction forces. I'll come to this in a minute. And there is also the bulk flows in the cytoplasm of the fluid with the divergence condition. And the divergence condition and the flow at the boundary is given by the surface flows. This extra term comes because if you try to write down flow equations on manifolds, curved surfaces, bit of differential geometry, you will find that there is something called the Gaussian curvature, which automatically couples to vector fields defined on curved surfaces. So therefore, you need to write down the appropriate covariant derivatives. These are all surface covariant derivatives. Sorry, these ones, and these ones too. And so the covariant derivatives on a curved manifold don't commute, and the difference is basically the curvature. Anyway, so the point is that you can establish a picture where you have a surface, something on the a cell, something on the surface, generating active stress, active flows, which drives bulk flows. Bulk flows get feedback into the surface. And what you get is something very nice. So it turns out the essential thing seems to be one parameter, which is basically the ratio of the shear viscosity of the cortex to the cytoplasmic shear viscosity, shear viscosity of the cortex. And that is a length scale. You compare that length scale with the radius of the sphere, and you get some very nice instabilities. So it turns out that you can get a growth rate, which can be such that you have a L equal to 1 growth, which corresponds to a single spot, or a L equal to 2 growth, which corresponds to a ring. So basically, if you have this length scale being much, much greater than L naught, you get a spot-like feature. If this length scale is much less than R naught, you get a ring-like picture. So pictorially, that means that you can get a pattern which looks like there's one big spot. There's only one big spot. That's like a cell polarity pattern. There's something here, nothing on the other side. Or you can get a ring-like pattern. That's a cell division pattern. So one of the earliest pictures that I showed you is to think about the cell and think about the pattern formation in the actomyosin cortex of the cell, which actually forms a ring and pinches the cell. So it's a very toy model, very simple toy model, which can give you a very nice understanding for how a cell division pattern can come up because of the coupling between surface stresses, bulk flows, surface flows, and natural couplings between them because of the geometry. Okay. All right. So it might be getting a bit too much. I want to give you a flavor for the kind of problems that are there in biology and the kind of problems that are very useful to think about, where the tools that you learn then can really be applied there. So, yeah. The question is, what is this last picture? So what you're seeing here is the concentration field on the surface with the surface velocities. And here you have a picture, it's a slice of the flow in the middle of the cell, in the interior of the cell. So this one drives 2 vortices, this one will drive 4 vortices. This is a plot of the vorticity, for example. So you can see this structure. So this one is like 2 ring-like things coming up there. So this is a sphere, full 3D. So the flow, this is only a cross section. This is a cut of the flow. This one is showing the full isocontours of the vorticity. I think it's the vorticity, but it might be the magnitude of the flow. I don't remember. All right. So if you remember, typical process of cell division is the following, that a cell typically rounds up just before it wants to divide. And what it does is to form a ring-like pattern of these actomyosin force-generating units on the center. These ring-like patterns, you can just think of it like a rubber balloon with a rubber band coming in the middle, and the rubber band has been stretched, so the rubber band tries to pinch it. So the next process is basically something where the cell undergoes a shape like that. This is really the ring pattern that's there. And so it's a natural question to ask, how is it that a cell divides? And so here's an example where the division pattern that comes up here naturally, like a ring, well, surely it will have an active stress there. And if you make the surface deformable, this should lead to a division-like pattern coming up. Yeah, I hope Aditya will tell me that in another 3-4 years. He's doing that story. Yeah. So it becomes harder now. Now, not just deformed curved surfaces, but you have to worry about deformable curved surfaces. You can write down the math, but as you can think, computing anything is very, very non-trivial. And so we can do things numerically, semi-numerically. Okay. I think I lost the share. Did I lose the share, or I don't know what you can see? Can people see the sharing, the screens, the slides online?
Yes.
Okay, great. All right. Okay, so this is a toy model for cell polarity and also for cell division. But it's a model which explains things nicely. So I thought by this time it would have become too much and most of you would have gone to sleep. So I put a nice video here to wake you up. One of the things that is very puzzling to understand, like I mentioned earlier, is to understand how organisms develop their body axis. How do they know their left and right? How do they know their front and back and head and tail? And how do they generate these axes? How do they generate shape? One primary axis that is established in almost all creatures is the head-tail axis. That's typically the first axis that is established in all creatures. In this creature, C. elegans, turns out that that particular axis is established by a certain cell polarity pattern that appears on the surface of the cell. I've shown you some pictures of that, so I want to run through a couple of slides to show you how this can be modeled quite quantitatively. So before that, a video to warm you up. So this creature is a nice worm, few millimeters in size, and it's more or less transparent to white light, so you can see through all its internal organs. Turns out that it has a nice embryo. Can we dim the lights here? The IT people are around. Dim the lights here. And maybe even this one. I don't know if there's a switch for that. There's a switch for this. Yeah. So this is a creature which has a whole assembly line of eggs in the inside of the creature. And you can take out one such egg and put it under a microscope. This is one egg. So it's about 50 microns in size, roughly, the long direction, more or less an ellipsoid. It's encased in an eggshell. You can see the thin outline there. And so if you keep the temperature pH similar to what is there in mummy's belly, it doesn't even know that it's outside. So you put it under a microscope and you watch it. And this is a marvelous process of development. So it starts out by being a single cell, just about to divide into two cells. You see the two nuclei there. Run the movie, and you see 2 cell, 4, 8, 16, so many cells dividing up. And eventually, on a time scale of a few hours, roughly around 11 hours, you'll see a full worm coming out. So here you see a huge number of cells that divided. The cells start rearranging themselves into a tubular shape. A worm wriggles around a lot. Meanwhile, it's developing all its internal organ systems. It's still encased in an eggshell. In about 11 hours, it breaks open the eggshell and comes out. So you just started with a bag of chemicals which is 50 microns thick. And 11 hours later, you can make a worm. Well, life knows how to make it. We don't know the physics of that yet. We are very far away from this. Anyway, first things first. The very first cell division of this creature, the same movie, very first cell division, turns out to be an asymmetric cell division in the sense that there are 2 daughter cells which are asymmetric in size. This asymmetry is driven by a surface polarity pattern. Polarity pattern in 2 chemicals, red and blue, you're looking at the surface. And at the same time, these large-scale cortical flows, the flows that for which I showed you the modeling. So this polarity pattern of red and blue tells you where to cut the cell into 2 pieces, basically the interface between the red and blue. And that's how you decide which daughter cell becomes bigger, which daughter cell becomes smaller. Some chemistry about this. You don't need to worry about the molecules and so on. So the essential crux of the matter turns out to be that there is a surface bound fraction of these proteins, and there is a bulk fraction of these proteins, and the total amount seems to be a constant, very similar to what I wrote here. Okay. And so there's some exchange between the surface and the bulk. Okay. There's some complicated chemistry. We don't need to go into the details. The essential idea turns out to be that A and P, these 2 proteins, the red and blue proteins, don't like to be around each other. So if there is more of A, it tries to remove P on the surface. And if there is more of P, it tries to remove A. So it gives you some antagonistic interactions. In the dynamical systems language, this basically means it gives rise to bistability. So there can be states which are A-rich, or there can be states which are P-rich. So what I'm plotting here is basically the null lines. So for example, if you have a 2-species model, plot the 0 contours of the chemical reactions, and you see a system where there are 3 fixed points. The middle one is unstable. The 2 black dots are the stable fixed points. So remember, go back to your first lecture that we had. We had a case where there was x equal to 0. It lost stability. 2 more fixed points came up. Which one is stable? Both are stable. Now, if you do a numerical simulation, which one will you go to? Like we did in the tutorial, depends on the initial conditions. So there is a basin of attraction for each one of them. And depending on where your initial condition is, you can go to this one or that one. Similarly, you have a case where there are 3 fixed points. 1 is unstable, 2 are stable. Which one you go to depends on the initial conditions. These are homogeneous fixed points. I can ask for the stability of such systems. Okay. So this is just a generic theory for the same model. I'll not go into that. So the general idea seems to be the following. You can put in some simple chemistry, for which we know evidence from experimental measurements. And the idea seems to be the following. You can have systems which are homogeneous states are stable. At some point, they lose the homogeneous state. The stability is lost for the homogeneous state. And you can get a pattern state, a high state and a P high state and so on. So there is a phase diagram. So the phase diagram is saying for the homogeneous state, there are 2 states. There is a blue rich phase, a poor state, or a red rich phase and a blue poor state. If you give in spatial degrees of freedom, like diffusion here, you can, in addition, to get patterns. And there seems to be a further bistability that you have regions of parameter space where homogeneous states and pattern states coexist. So go back to your first lecture, we had plus root r and minus root r, and they're both stable at the same parameter value. So in principle, some of the questions that came up about nonlinear stability. I mean, suppose the perturbation that you put is not small enough. You actually put it a large enough perturbation that it goes from one stable state to a, of course you can do that. But analytically, it becomes very difficult to prove anything there. And this is an example where such a system is seen in a real life experiment. So you have a system where a homogeneous state and the pattern state are both stable at the same parameter values. And so what's happening is the following. Our claim was that this transition that you saw from an unpolarized cell to a polarized cell is basically a transition from here to here at the same parameter values, driven by a perturbation which comes externally. It's large enough perturbation so that it's linearly stable. Linear stability answers value tell you it's stable, but you have to put in a perturbation that will take you over the basin of attraction. Okay, so now it goes into a bit of biology, and I want to give you a feel for the physics, that's all, and you can forget the biology. So I said that both these states are locally stable. So that means, let's say I take this state, I put in a perturbation as seen in this movie, and you see the system tries to form some pattern, but then if I remove that perturbation, okay, it doesn't go through, it just comes back, it's locally stable. Put in a perturbation which exceeds a certain threshold, which depends on all the parameter values. This has to be determined numerically, and the system goes over to a polarized state and it stays there. So this is stable, that is stable, and I can go from here to here by some signal. Okay, I will rush through this. I don't want to go into too much details. So naturally the question is, in the biology, what is the signal? So that's where the biology comes. Turns out that there are two signals that come up. I will not go into the details. And we can model the whole system by a set of equations which is very similar to what we wrote down for these species. So right now you have two species which form the red and blue proteins, and you have a species which regulates the active stress. If you recognize these equations, I've done my job. So it's very similar to the picture that we had today morning and yesterday in yesterday's lecture. And this couples to certain pattern forming species. The experimental collaborators, we were able to get very good data. So that means we can take movies like this and make experimental data plots like that. So naturally, if you have a theory for this system, there's a natural question, well, can you actually use the theory to compare with the experimental data? Turns out the answer seems to be yes. There's a large number of parameters, but we've done a systematic elimination of parameters. I'll just flash through things. Turns out that you can do the system piece by piece. You can take apart the chemistry alone, the mechanics alone. The solid lines are the fit from the the theoretical model, and the points are the experimental data. Remember, all this is inside a living worm. And so you can actually predict the patterns very, very quantitatively. So you can do a complementary thing where you knock off the chemistry, and you can get something which there's a complete theoretical prediction of the whole pattern. Okay, so my idea was not to give you a feel for how this worked and all the details of that. That was not at all the point. The point was just to show the kind of tools that we used to understand simple reaction diffusion patterns, patterns that undergo stability, they lose their stability when you encounter a bifurcation. Similar ideas can be used for looking at real biological patterns. So take home picture that I said, that there is a fixed point, which is a homogeneous fixed point. And there is a fixed point, which is a pattern fixed point. And there are basins of attraction between the two. And there is a separatrix. So if you have a signal that takes you from the homogeneous fixed point and puts it into the basin of attraction of the inhomogeneous fixed point, well, that's enough. Then the system will automatically flow towards that fixed point. And we showed that that is what underlies this cell polarity picture. And so we were able to measure many things and show you and basically map out the complete phase diagram. Okay, so I want to spend the last few minutes on some really open problems, and I will end there and take some questions. No, wait, what happened to this? No, not this. Okay. So I'll leave this slide and let's discuss what are the front tires. So far, I've been talking to you about patterns which happen on a fixed domain, maybe in one dimension, maybe in two-dimensional surfaces, and maybe towards the end on a sphere as well. But these are all patterns which are fixed on a fixed domain. The shape of the domain is fixed. But surely biology is not about fixed shapes. If you look at all of us, we are not spheres. So therefore, if you just keep solving things on spheres, then you're never going to get to the real biological problems. So you would like to understand, in addition to patterns in, say, concentration fields, orientation fields, maybe stress fields, all these density fields, all these patterns in these fields, you also want to understand patterns in geometry, in shape. So one would naturally want to ask questions about how is it that living systems generate shape patterns? How do they shape up? And probably the simplest kind of example of non-trivial shape is a dividing cell. So you had patterns in, let's say, concentration fields, maybe velocity fields, maybe orientation fields, and so on and so forth. And we developed a description which could be a simple reaction diffusion picture without any mechanics, or an active hydrodynamic picture which involved forces and stresses, and so naturally had couplings to velocity fields and orientation fields. But these are all on a fixed domain. So you did it on a shape, did it on a domain which was fixed in shape and size. So maybe you had a one-dimensional domain, maybe you had a square domain, or maybe if you're brave enough, you did it on some shape which was fixed. So this itself has a huge number of unsolved problems, in particular about how shape controls patterns. Let's say you fix the shape, how curvature affects patterns. Can curvature lead to positioning of patterns? Can regions of high curvature attract concentration fields? Can regions of low curvature repel velocity? We don't know. So couplings between shape and pattern is something which is a really open problem. One would like to understand that. Well, this is, if the shape is fixed, you can do something. I mean, you can at least write down the equations reasonably easily. The equations are easy to write down, but almost impossible to solve, because the nonlinearities that come from geometry make it very, very hard. So geometric nonlinearities are very hard. But that's not the real problem. I mean, the real problem is to go to a case where you have a shape which is dynamical and a pattern that is also dynamical. Well, I mean, shape is also a geometrical pattern, but when you say a pattern, maybe you mean in terms of these. How would one write down a consistent theory for these kind of couplings? I mean, if you just stare at it for a few seconds, you should realize this is basically geometrodynamics. Where fields like the concentration field, the velocity field, the polarity field, the orientation field, many other things, these enter active stresses. Active stresses generate flows and deformations. Flows and deformations change shape. Shapes will affect the kind of patterns. So you really like to understand what are the minimalistic kind of couplings, minimalistic kind of theories, which will probably give you some understanding of the kind of shapes. Simplest pictures that you can think about, and this is something people have been working on. So probably one of the first things to work out this and show some non-trivial dynamics is this paper. These authors basically take the same simple single species active stress regulator model, put it on a surface, make the surface a fluid-like surface, and they start looking at the kind of shapes the surface takes. There's a plethora of papers coming up now in the past 3-4 years where one would like to understand these kind of patterns. So simplest examples, if I have a sphere, think of the cell as a sphere, I showed you that by coupling to the cytoplasm or maybe some other ways, you can get a pattern where there is a high concentration of some chemical on the surface. Now, if the sphere were deformable, you would obviously get a dent in that point, because a concentration pattern also generates a stress pattern. And if you have a deformable surface, you go back to your simple elasticity theory, for instance, a stress pattern will change the shape. Now, this will maybe enhance the concentration fields, maybe deplete the concentration fields. One doesn't know that. Would like to work out these kinds of shapes. So the thing that I want to leave you with is that geometry is also a shape pattern, and trying to understand that in the language of the tools that we've been developing is a hard open problem, which would probably give you a good understanding of many shape patterns in biology. Okay, so I think that's good enough. If there are questions, comments, that will be good, and I'll stop there.
No, okay. Any questions from the online people? No, okay. So, yeah, if there are no other questions, then let's thank Vijay for a really wonderful set of lectures. So, yeah, thanks.
Hope it was useful and fun. All right.
Yeah, thank you, Vijay.
Yeah.
Thank you. Thanks, everyone. So if there are questions, comments, if you want to say something, you can also drop me an email and I'll try to respond whenever I can.
Vijay, are there any like handwritten notes or something that you can?
Yeah, I've not finished it. So I mean, not this part, but the initial parts, I will circulate some notes.
I mean, we can put them on the website.
Yes. Yes.
Sir,
Yeah.
I have a question here. So here you said that a different shape. Yeah, so you said that this coupling between the shape and pattern, that still people are struggling to find. But what about the dimension there? Like
Dimension in the sense?
Two-dimensional objects like.
Yeah, surely. I mean, you can start from one-dimensional things. Plants grow. So you can start from one-dimensional lines which are growing. So you can think of in one dimension, then there's nothing but growth. And troubles about when does it know when to stop. Okay. Well, let's say you make it one-dimensional object sitting in 2 dimensions or 3 dimensions. It can turn. Haven't you seen twigs and all? So there are questions about how is it that they know to turn, to shape. You can look at surfaces embedded in 2D. Cell surface is an example. The cell, rather than being a sphere, sometimes becomes an ellipsoid, sometimes becomes flattish, gets polarized, gets divides. How do these shapes come about? More complicated things. You have a bunch of cells put together, a tissue, which is really a 3-dimensional aggregate, or could even be a 2-dimensional surface of tissues, epithelial skin tissues. You get folds. Think of your brain, think of your guts, your lungs. How do these shapes come about? When an organism develops starting from an embryo, you don't get all these shapes. It's not like all these shapes are tiny things and they become big. There's nothing there. It's basically a single cell, much like water. So these shapes spontaneously get patterned. How do they do that? Obviously, it's not just chemistry, because there are not just molecules going up and down, but there are also shape transformations. stress, there's mechanics going on. Must be a strong coupling between active mechanics, chemistry, and something which I have not even touched here, signaling. Different parts of the organism talk to each other, different parts of the cell talk to each other. And this is something which is explored a lot in biology. A huge amount is known, but we don't know how they couple to shapes, how they couple to mechanics, how these three things talk to each other. We don't know all those things yet. We're very far from it.
And one more thing is pH is there in the cells, like pH regulates a lot of things in the cells. So in the differentiation,
pH, temperature, many things. So again, simplistic models where if you start from in biology, what happened to that? What about this? What about that? Well, there are a million things so we don't understand all. So what we are trying to write down are simplistic descriptions. Maybe if you neglect all that and see if you can understand this part, maybe this part, maybe that part, maybe this part, depends on the question that you're asking. Also on the length and the time scales that you're looking at. pH regulation is quite fast. This one is slow. So maybe on those time scales, you can forget that.
Okay. Yeah. Thank you, sir.
Yeah.
Is the coupling between the concentration pattern and the active stresses a one-way coupling, or is there a two-way coupling as well? I mean, does the active stress in turn again affect the concentration pattern? Is there a feedback loop, or is it just a one-way?
Surely it's a two-way feedback loop, right? Because the models that I showed you, there is a concentration field and maybe some orientation fields, and they generate the stresses. The stresses will generate typically flows and deformations, which will affect the concentration fields. So that's the simple model that I worked on. Now coming to shape, if the stresses are generated, you know that a stress on a deformable material, well, by definition, deforms the material. So once the material gets deformed, naturally the shape of the material will feed into the kind of patterns that will come. Does that make sense? So the patterns get affected by the shape affects the patterns.
Yeah, so you also talked about the cell signaling part.
Yes.
Right? So that has been studied a lot, right?
No. I mean, that has been studied a lot on fixed shapes. I mean, never with mechanics much. It's most of the studies have been on chemistry. I mean, a large part of our work has been really to understand even the signaling mechanisms. What are the molecules involved? How do they talk to each other? And which molecule regulates what kind of processes in cells? It's a huge field. So the coupling to mechanics shape is barely touched.
What about the cardiac mechanics side?
What? Repeat that.
Cardiac mechanics. I mean, the coupling between the electrophysiology and the mechanics of the heart.
I didn't get that. What mechanics?
Cardiac
with the heart. Yeah, of course. I mean, that's a much more complex system. I was just thinking of a single cell. If I can understand a single cell before I die, that'll be great. Forget about the heart. It's too far away.
All right.
Yeah, I mean, one can, of course, write down more complex, more coarse-grained models. People have studied, for example, ventricular fibrillation and how scroll waves and spiral waves are generated in the heart, voltage patterns that come up there. Couplings to how the heart functions by beating and shaping and pushing flow, quite far away. Question or comment? Thank you. Okay, fine.